So we made it through the ovary, we made it through the uterine tube, and now we are on the uterus. And it is anterior to the rectum, yep, posterior to the bladder, and slightly superior to that. And it is anchored in place by the broad ligament. And you already have notes similar to that. One of the things I do want to point out to you guys, these things that they're calling ligaments, they're actually mesentery, because if you take a look at some of the cartoon drawings, it shows you that it is like a membrane and a membrane applied. So we just unfortunately have given them the name ligament because it's an obvious structure that they see, and various other things are also encased in those. Now the uterus, the bulk of the uterus is the body of the uterus, and take a look at the cartoon depiction, and indeed it's fairly obvious to that. But there is an area called the fundus, and we had fundus of the stomach, and remember the fundus was the rounded most superior projection of the stomach. Same thing here with the uterus. The fundus of the uterus is the rounded most superior projection of the uterus itself. The cervix, our notes here state it has a narrow outlet. We also call the cervix the neck of the uterus itself. It's obviously inferiorly positioned. And we have internal plus external osses. Now an os is an opening, so it's Latin for opening. You may have taken medical terminology, for example, PO, per os. And that typical opening that they're referring to is the oral cavity, but this is the cervical opening here. Internal and external os with a canal, cervical canal, in between. And one of these openings is actually where a pap smear is done. So which opening, which os is where a pap smear is actually done? See, external os. Now let's go up to our cartoon picture here and take a look at this, because the reason pap smears are done is it's for cervical cancer screening. And so what is it about this area that will make it susceptible to cancer? And it has everything to do with the mucosa here and the mucosa here. So the mucosa inside the uterine body and a portion of the canal itself is a typical mucosa that you think of. We have some columnar cells that are attached to a lamina propria, and it's going to have various aspects of vascularity because it's a very vascular mucosa that the endometrium is actually made out of. But it's an endometrial lining that's very, very glandular. And it's very glandular if you take a look at the cartoon itself and you follow it into the canal. It's glandular all the way through the canal, and hopefully you can appreciate that thickness that they try to depict with this starts to diminish. And that's because now we're meeting an area that is totally different as far as the cellularity of the epithelium. And this cellularity of this epithelium should make sense to you because it is in an area that is going to undergo trauma and friction. And what type of epithelium is best for that? Stratified squamous epithelium, and absolutely that's what it is. So we have a transition zone between glandular epithelium and stratified squamous epithelium, and that makes that transition zone susceptible to pathogenic invasion by various viruses to cause actual cancer. And so pap smears are done in this area because it's designed to get that transitional area for you. Now, the other thing about the cervix is we have the external and internal os, and we have this canal in between. I'll go ahead and erase that stuff. But the canal is glandular, and that glandular aspect is very important because glands produce what? Some sort of secretion, right? And so these glands are going to be mucus-secreting glands, and they're going to secrete a lot of mucus, and this mucus is very important because it creates a plug. And why do we need a plug here? Because the outside world is in contact with the peritoneal cavity of the female's body. Think about this. Entry in through the vagina. Vagina is kind of in contact with the outside world, correct? Look at this cervical canal. The osses are still open. You have to realize that these are openings. That's what an os is, an opening. So we have an external os and an internal os, and we have the cervical canal in between, which is also a space. Then you have the uterine cavity. Then you have the interior of the uterine tubes. And then if you are some sort of pathogenic microorganism, then you go out and you do a swan dive into the peritoneal cavity. So this cervical canal is actually the area where we need to make sure that there is a plug, and that's what the cervical canal is all about. It creates a mucus plug that keeps things from actually penetrating into the uterine cavity and then wreaking havoc with the reproductive system and other structures of the female. So it seems like a kind of a trivial thing to take a look at here, but this is a very important entry portal. And we will discuss the importance of this entry portal whenever we discuss conception, because in order for conception to occur, we have to navigate various barriers, and this is a very important barrier for that. Okay, let's go back down to the notes portion here. And wall of the uterus, perimetrium. Perimetrium is a misnomer, and I hope you realize a misnomer means it was misnamed, okay? And because it's actually just visceral peritoneum. That's what perimetrium is, just the visceral peritoneum layer. And we know that's a serous membrane. Then we actually take a look at deeper, going from the surface to the deeper area. We have myometrium. This is a very thick muscular layer. And then we have endometrium, which is indeed a mucosal lining. So we have some nice glandular type of cells. The columnar cells and cuboidal cells are obviously very nice and glandular. But the endometrium is interesting in that it is partitioned. It has two strata, two areas that are designed to keep the endometrium itself healthy, because you may realize that the endometrium changes throughout a menstrual cycle, and part of it is actually sloughed or lost during menstruation. So which one of these two layers is, as you take a look at the names, would be the one to be sloughed? Stratum functionalis, absolutely. That is the functional layer that is shed during menstruation. And I'm going to say menses because it's so much shorter to write than menstruation. Okay, so that layer is sloughed or shed during menses, and so it needs to be built back up, and it's easy to build that area back up because we then have the stratum basalis, which is the basal layer that will replace the lost stratum functionalis. And so if you have the stratum basalis, it is next to the myometrium. Stratum functionalis is next to the actual uterine cavity itself. The other thing to note is the uterus intraperitoneal or retroperitoneal? Intraperitoneal. So you can take a look up there and realize that we actually have it protruding up in there, but then we go into an area that is no longer intraperitoneal because we have the term adventitia associated with that. So that's the vagina, also referred to as the birth canal. 3 layers to it, or tunics. Adventitia is fibroelastic, allowing it to obviously have some distension. And because we have adventitia now, you have to realize we are retroperitoneal. The vagina also has a muscular layer. It's actually 2 layers of smooth muscle. And the mucosa, as we actually work through, is indeed stratified squamous epithelium, and it's organized into rugae. And you guys know what rugae is for, correct? It's for distension. So when it's not distended, you have rugae, and when it is distended, you do not have rugae. Now, this mucosa is going to be actually responsible for producing some acidic secretions. And actually can get that pH down to 3.5 or 4.0. And that's important for what reason? Yeah, it actually is going to inhibit bacterial growth, and any foreign material that could actually enter into that area can actually have a negative or deleterious effect on that. Moving down to the next topic, external genitalia, also referred to as vulva. The mons pubis is indeed some fatty tissue over the symphysis pubis that actually provides some cushioning or padding to that area. Then we have labia majora and labia minora. Labia majora, as the name says, it's the larger of the two, and they are the most obvious 2 skin folds running from an anterior to posterior aspect along the area called the perineum of the woman, and the anterior aspect would actually start at the mons pubis. It is analogous to the male scrotum. The labia minora are very different from the majora. They are actually mucosal folds, not skin folds, but mucosal folds that enclose an area called the vestibule. And the vestibule is, if you remember the ear, the inner ear, the vestibule is an area, a space, and so that's what we're talking about here. It encloses a space where we will actually find various structures. In the most anterior aspect of that, we'll find the clitoris. We'll then find the urethra. Then we'll go find the actual vaginal opening. And then most posteriorly, we'll actually find some glands. There are bulbourethral glands in the male, and so the female has kind of an analogous type of glands, but you don't have to know the name other than their glands. Very similar, and when we get into the male system, you'll see what the bulbourethral glands actually produce. In the woman, they're called Bartholin glands, and those actually can become infected. The clitoris is true erectile tissue, and it is analogous to the glans penis of the male. Moving to some other secondary sex characteristics or organs that the woman has. Mammary glands, indeed functional only in the female, in that those are the women are the only ones that are able to actually produce milk because of various hormonal features that the woman actually has. What's interesting is that they are actually modified sweat glands. And so actually belong to the integumentary system. Some features I want you to know about with the mammary glands. The area that surrounds the nipple is called the areola, and so it is indeed pigmented skin. The breast tissue is actually subdivided by connective tissue septations or walls into lobes, and each gland will contain anywhere from 15 to 25 lobes. And so the CT, which helps to separate the glands into the lobular tissue, also then has extensions at the base of the breast that actually attach it or keep it applied to the muscles of the chest. And so what muscle is the mammary tissue actually attached to? The pectoralis major. And those CT extensions are called suspensory ligaments. When we go into the actual reproduction chapter, we'll talk about how these actually change into actually functional milk-producing units. The physiology of the female reproductive system is a bit complex. And the process of forming the gamete or the egg or the oocyte or oocyte, however you want to pronounce it is actually fine, is a process called oogenesis. And we want to make an egg. We want this guy that will get fertilized by the sperm to form the zygote. Another term that I will use for fertilized egg, besides zygote, is conceptus, because conceptus is a product of conception, and it's a real super generic term. So once it's fertilized, you can call it conceptus all the way till it's born. But if you're not quite sure what term to use. But initially, when it's fertilized, we call it a zygote. And as we will go through the next chapter, we'll assign other names to various stages of development. But conceptus is a very generic term. So let's take a look at this thing called oogenesis, development of oocytes. Now, take a look here at this blue stuff. This is before birth, embryonic and fetal period. What happens is each ovary have its own type of stem cell or germ cell, and they're called oogonium. So the oogonium or oogonium are stem cells, and take a look at the chromosomal number. This is very, very important. It's 46. So how many sets of chromosomes is that? 2 sets. And this cell will undergo mitosis until the ovaries are adequately populated with the egg precursors. So these guys, as the notes say, undergo mitosis. So they're mitotically active, which means when they undergo mitosis, you have 2 exact copies of the original, correct? And so they will do this until the ovaries are adequately populated with millions of these oogonia. Then, once the population has been achieved for the ovaries, then these cells are no longer mitotically active. This is very different in the male system. So what you have to realize, we have mitosis in ovaries for women, we have mitosis in testes for men, and that will always give you, with mitosis, 2 sets of chromosomes in the offspring cells, or the daughter cells. But the mitosis process stops for women. The mitosis process does not stop for men. And when the mitosis process stops because of adequate oogonium population of the ovaries, they will then switch into a gamete-producing program called meiosis. Meiosis has 2 phases to it. We have meiosis 1 and we have meiosis 2. Now, meiosis 1 and 2 both have very similar stages that we saw with mitosis. Do you guys remember the stages of mitosis? The first one was called prophase, metaphase, anaphase, and telophase. So in meiosis 1, you have prophase 1, metaphase 1, anaphase 1, telophase 1. Same process with meiosis 2. So meiosis is different from mitosis in that the outcome is going to be gametes. And do gametes look the same as what I call the mother cell? They do not. The mother cell has how many sets of chromosomes? 2. The gamete will have 1. So the gamete will look very different from the mother cell. And In this process, what's really interesting is this is where we'll have, in meiosis 1, we'll have what's called we'll have reduction of chromosomes, chromosome sets, let's see here, in 1. And in 2, we will have equatorial division. Do you remember in mitosis, we line up, when we get to metaphase, we line everything up on what's called the metaphase plate, and we then pull apart the chromatids and take them to separate poles. That's what equatorial division is actually there. And reduction in chromosome sets means that we're going to go into what's called haploid. And in equatorial division, we'll still produce haploid cells. So haploid means 23 chromos for both areas, but it's by a very different creative way of looking at things. And if you want to know more about meiosis 1 and 2, I actually suggest you get out there and look at some good YouTubes for that. But if you just want to keep it simple, let's go ahead and that's all I'm going to tell you about it. And let's go ahead and take a look at this chart now. So oogonium is a mitotically active process until we get enough, and then we are destined to become primary oocytes. So take a look here, primary oocyte, and this is still in utero. So this is still within the womb before the child is born. We are now, if you are in prophase 1, I want you to realize that's meiosis 1, because we don't say prophase 1 for a mitosis process. So this is meiosis here. Prophase 1 means we've started meiosis 1. When you finish meiosis 1 and go into meiosis 2, you will be a secondary oocyte. So primary oocytes are going in, that are part, basically have entered meiosis 1. When they finish meiosis 1, they will be called a secondary oocyte and will go into meiosis 2. With this process in the male reproductive system, we will see from one mother cell, we will end up with 4 gametes. In the female system, we go from one mother cell to 1 gamete. So we lose gametes along the way. And let's take a look at how this happens. So before a girl is born, she has all of her eggs that she's ever, populating her ovaries that she's ever going to have. And she goes into the gamete producing stage of meiosis 1 and gets arrested in prophase 1. So this is a state of suspended animation. This is like one of your sci-fi movies where people travel, many light years away, and they put them in these pods and they put them into a deep sleep, the suspended animation, and they don't age at all. Well, they never really show you that not everybody lives through that process. And that's what's important here. So all of these eggs in the ovaries are now put into meiosis 1. They're in a prophase 1 in suspended animation, and we're going to actually have some loss. And so we populate the ovaries with a whole bunch of pre-eggs, and then we put them into suspended animation for years. When do we start taking them out of suspended animation? Puberty. So for years, they are arrested in prophase 1. So all through childhood, there is attrition. There are actual loss of some of these pre-eggs, is what we'll call them. Okay. Now puberty starts right here. And from puberty to menopause, what will happen is women will actually experience recruitment, monthly periodic recruitment to actually complete meiosis 1. And so here we have, it says primary oocyte arrested in prophase 1, And you see now, you see the term meiosis II arrested. Well, in order for meiosis II to be arrested, that means we had to complete meiosis I. When we complete meiosis I, we go from 46 chromosomes to 2 daughter cells with 23 chromosomes. What I want you to tell me is, what is the most obvious discrepancy between these 2 daughter cells right here? Size. Absolutely. One is literally the chromosomes, and the other is chromosomes and cytoplasm and everything that you need to survive a very long camping trip. So that's why this one right here, this one right here, the largest one, is large. Because with cytoplasm, you know, all the machinery that a cell needs to do its job or to live or survive is in that cytoplasm. The other one is just a bunch of chromosomes. So let's talk about the polar body, which is a bunch of chromosomes and nothing to sustain it. Say actually somehow a sperm got to it and fertilized it. Would that thing live? No. So it is actually designed to not live. I want you guys to realize the polar bodies are designed to not live because we need the life-sustaining material of all that cell of the cytoplasm to go with the one that's designed to live. And so we have now our secondary oocyte. And remember, secondary oocyte means that we have completed meiosis I and we are into meiosis II. And guess what, guys and gals? Again, the girl is going to make you wait. So it will get arrested in metaphase II. So that's telling you in meiosis II, we also have a pause. And the pause is nowhere near as long as the pause that was in meiosis I. But meiosis, this is why I make fun of us. You know, I like to make fun of human beings, male and female. I mean, like, I love to not discriminate against who I make fun of. But when guys want to take a girl on a camping trip, they say, you know, honey, you don't need to pack all that stuff. And they go, oh, yes, I do. And they are right. They are right from even a cellular level. They have learned this lesson. From even before they were formed, they have learned this lesson. So guys, quit arguing with them and just go with it, you know, kind of stuff. You can't tell me you haven't learned this already, you know, so. So we're in meiosis II, and this is the oocyte that's going to be ovulated. And so this nice, big, healthy gal is going to be ovulated. And when it's ovulated, it becomes the ovum, because ovum and ovulation are kind of synonymous then. And it's still stuck in meiosis II. And she isn't going to get ready and complete meiosis II until her date's at the door, knocking at the door, literally. Because that's another thing that you may have noted about girls. When you take them out on a date, you have to wait. You get there and you wait because they're not going to finish getting ready until you actually show up. Not all women are that way, but a lot of women are. And still, to me, the story is the same, you know, on a cellular level. So ovulation occurs. It gets into the duct system, the uterine tube. Remember, fertilization is supposed to occur in the ampulla of the uterine tube. And that's what our note here says. Meiosis II is completed if fertilization occurs. So the sperm is actually the signal for completion of meiosis II. Once meiosis II is completed, because now it's been completed and you have 2 packets of DNA, each set of 23 chromosomes, that fertilization then produces what we would call a zygote, also a conceptus, because that's the first thing that comes together. So you have to realize that here it calls it an ovum, and I prefer us to use the term ovum for ovulated egg. And then after fertilization occurs, I prefer you call it either conceptus or zygote in the very early stages. Does that make sense to you guys? You know, ovum, ovulation, and then conceptus means fertilization has occurred, and then, or else a zygote means fertilization has occurred. And so the other thing, too, when meiosis II is completed, note that we have another polar body. Again, this is not designed for survival. The one that is designed for survival is the one that completes meiosis II with the most cytoplasm. Because then it has to take a trip. It has to take a trip that lasts 2 weeks long before it starts to get its nutrients. And so this is indeed why the egg, the ovulated egg, and the resulting fertilized egg is such a large structure, because it has to go down the uterine tube to the uterine body into the cavity of the uterus and then undergo a process called implantation. And if it does not successfully implant, it means it's going to actually then be lost during menstruation, because if it cannot implant successfully, it cannot then promote the maintenance of the uterine lining. So even if an egg gets fertilized, it is not guaranteeing that it is actually going to become an infant unless it undergoes the next stage, which is successful implantation. And so you have to have all of this cytoplasm and all of this stuff coming along with this fertilized egg that the gal is bringing along, because sperm cells are basically just a packet of DNA and some digestive enzymes to help it get to the egg to actually go ahead and inject that DNA into it. And so when you're just carrying a packet of DNA, you're not carrying any real cytoplasm. And so 2 weeks of survival for a polar body fusing with actually a sperm cell guarantees actually no survival. So this is a process that was designed to maximize survival of successful fertilization. So that's an important process to realize. So we talked about a lot here about this line, the oogenesis process. Now let's just switch over to the right-hand side of this and take a look at what is happening at the same time in the ovary, okay? So we see here, remember, oocyte is the thing that's inside the follicle, so the very center thing where DNA is located. And if it's a primordial follicle, that means that it's simple squamous epithelial cells that aren't doing much other than hanging around. Well, now that we have certain hormones telling things to happen, take a look at what happens in the ovary whenever we actually are starting the cycle. We have a primary follicle forming, and that's the histology view of that. Remember, primary follicle means no longer follicle cells, but granulosa cells around that. Granulosa cells are producing the hormone estrogen. So little granulosa cells means little estrogen level. Lots of granulosa cells means high estrogen level. And that's what happens. We undergo a lot of mitosis. We become a secondary follicle, and we have more granulosa cells. We have more estrogen levels increasing until we are a mature follicle here, which shows it there, which is also, remember, called a vesicular follicle because it looks like a vesicle or a blister on the surface of the ovary. And around that egg, we have all of this fluid, but we still have a layer of granulosa cells around that called the corona radiata. She's got her guys because once she's ovulated, which is what this shows here, the corona radiata goes with her. This is her entourage. This is her protection, because things are going to be encountered, and she needs to be protected from things she shouldn't encounter. And the things that she should encounter for fertilization or sperm cells, they know how to navigate through the corona radiata to help with successful fertilization. So now we have, after fertilization, this is all ovarian on this side, ovarian. Now we have, after ovulation, we now have the formation of all of those granulosa cells into this special structure called the corpus luteum. Granulosa cells, you hear granulosa cells, you think estrogen producers. Corpus luteum, I want you to think progesterone dominance. But did I say no estrogen? I didn't say no estrogen, I said progesterone dominance. And that's what the corpus luteum is for, because progesterone is what maintains the uterine lining for successful implantation. If you don't have adequate progesterone levels, you will not have an adequate uterine lining to help promote successful implantation. So all of these things are vitally important for actual pregnancy to occur. The corpus luteum will have a peak progesterone production that we'll take a look at, and then it will start to decline. And if successful implantation doesn't occur, then the corpus luteum completely withers away into the corpus albicans. So we have on one side, we show what's happening to the oocyte. On the other side, we show what's happening within the ovary. And now let's go ahead and look a little bit more at our notes. Oogonia are, like spermatogonia in the male, primordial germ cells that are mitotically active. So that means we will go from 46 to 46 times 2 every time we undergo mitosis. And remember, the mitosis will occur in the embryonic period and the fetal period of the female until we have the proper population of oocytes, pre-eggs actually established. Then, during development, all go into meiosis. So we stop mitosis and we start meiosis 1. But we don't complete it, we get arrested in meiosis 1. And if it's easy to remember prophase 1 for you, that's great. But just remember, meiosis 1 gets put into a state of suspended animation. Each month after puberty, several primary oocytes are recruited to continue to develop, in other words, to actually complete meiosis 1. But typically only one makes it. And this is important because this is going to tell you about how many children you're going to have, correct? If we recruit several to start the race, but only one wins the race, that means you're going to have one child. But if you have several start the race and more than one completes the race, that means you're going to have 2, 3, 4, 5, 6. I don't know why anybody would want 8 children, but you're going to have a whole bunch of kids inside the womb developing potentially for you. So this process is actually typically designed for only one to actually be inside the uterus, because the more children you have in utero, the higher risk your pregnancy, which means the higher risk for complications for both mother as well as the babies. So if you do have more than one egg actually complete this process and complete meiosis 1, what does that mean about the type of twins you have, for example? They are fraternal twins because they come from 2 completely different eggs and therefore also 2 completely different sperm cells. So with meiosis 1 completing, remember, when that completes, we get our secondary oocyte plus our first polar body. Now remember, we are in the haploid area. Diploid means 2 sets of chromosomes. Haploid means 1 set of chromosomes. What's triploidy? 3 sets of chromosomes. Is that viable for human beings? No. In the plant world, you can have polyploidy, which is the triploidy and tetraploidy in the plant kingdom. In the human world, you can only be successful if you're a diploid. Now, that doesn't mean that you can't have an extra chromosome or 2, but I'm talking about complete sets of chromosomes. When you hear the term secondary oocyte, remember, it's going into meiosis 2, and it will also get arrested in meiosis 2, and will only complete meiosis 2 if fertilized. So if it's not fertilized, we just have a secondary oocyte that gets going to wander its way down the uterine tube into the uterine cavity and just wait until next menses and be sloughed and lost. Also at that time, because it cannot implant, we'll learn, there are actually macrophages inside the uterine cavity of women, and that makes sense since I just told you that it's connected to the outside world except for this nice cervical plug. And so you have other cells in there trying to actually protect you from pathogenic things that can actually enter in. Now, we're going to talk about this a little bit, and then we'll revisit this on next Monday, which, by the way, don't forget it's Pajama Monday. I'm wearing pajamas, so you won't be alone if you actually wear pajamas. So my yoga class goes, isn't this pajamas, you know, that we actually do every Monday and Wednesday, but I'll actually wear pajamas. So. Okay, so this here is showing us the ovarian cycle, showing us what's happening inside the ovary here. It's showing us what's happening hormonally here with FSH and LH, which are anterior pituitary hormones. It's showing us what's happening hormonally in the bloodstream. That's because of the units in the ovary producing these estrogen and progesterone, and it's showing us also what's happening inside the uterus with the endometrial lining. Day 1 is given its distinction because it's an observational event. You can say for every woman, whenever she starts her menses, that is day 1 of her cycle. This ovarian cycle that we're going to talk about, which has 3 phases in sequence in the ovary, is a 28-day cycle. Let me ask you, are all women 28-day cyclers? No. Some women cycle every 3 weeks, some 4 weeks, some 5, some 6, some 8, some 2, some 3, some 4 months. You know, and what that means is if they cycle every 3 to 4 months, they have menses every 3 to 4 months. Which one would you rather be? A woman that cycles every 3 weeks or every 3 months? 3 months. Most women don't view their menses as a very sociable event. So they just don't like it. And when it stops, that's why menopause is so great. When it stops, everybody that goes into menopause is typically pretty darn happy. So glad that thing went away is basically the way they view it. So take a look here at the top aspect here. This is based on a perfect 28-day cycle, and ovulation occurs right in the middle of that, so on day 14. So we have what's called the follicular phase, in other words, the development of follicles as the first portion of this ovarian cycle. Then we have a very short window for ovulation, and then we have the tail end, or the third phase, called the luteal phase, and it's called the luteal phase because it is dictated by the corpus luteum. So the follicular phase is the development of follicles. Ovulation then occurs, and then we aren't interested in developing any follicles. We're interested in priming the uterus for the possible implantation of a conceptus. So that's how it's subdivided. And when you take a look at it, we discussed these. Now let's go ahead and take a look at some hormone levels. And first we're going to look at hormones from the anterior pituitary, FSH and LH. FSH stands for follicle stimulating hormone. LH stands for luteinizing hormone. So let's see. On day 1, which is the beginning of menstruation, which is the loss of stratum functionalis, FSH levels are kind of high. LH levels, you know, they're not that high, but they're higher than what we see in the second half of the cycle. FSH is going to be responsible for, indeed, follicle stimulating hormone. So it stimulates, it says, okay, I'm the recruiter. I want this group of primary oocytes that are in suspended animation to join the game and join the race. And so we have these FSH levels starting a bunch of follicles to develop into primary follicles, which, remember, is going to start producing estrogen levels. So let's take a look down here at our estrogen levels and our progesterone levels. Progesterone levels are at all-time low at the beginning of menstruation. And that drop in progesterone is actually what brings menstruation about. Because progesterone maintains the endometrial lining. That's very important for you to realize. Progesterone maintains the endometrial lining. When progesterone levels drop, endometrial lining is not maintained, so then it's sloughed. Estrogen levels are also at their all-time low because we don't have any follicles developing those granulosa cell layers. But now, as we have follicles developing those granulosa cell layers, take a look, of course, what happens to the estrogen levels. They rise. So you know here at this top here, we have something that's got a lot of granulosa cells and it's producing a whole bunch of estrogen because of that. Look over here at what happens as the estrogen levels rise. What happens to FSH? It drops. FSH drops. FSH is the recruiter for the race. The race is running, and we already are in the midst of creating a winner. We don't want any more races starting. So FSH is the recruiter hormone. Okay, so FSH starts to drop as estrogens peak. And so it's a negative feedback. You guys get that? Increasing estrogens have a negative feedback on FSH release. Doesn't seem to have much of an effect on LH release. If you notice, LH is kind of just hanging out there. But what's really interesting, as estrogen peaks, instead of negative feedback, it suddenly has a positive feedback on FSH and LH. And that's what actually causes ovulation. This high peaking estrogen level suddenly, because it's now this kind of this max level, instead of negative feedback, it's going to have a positive feedback and a remarkable positive feedback on luteinizing hormone mostly, as well as FSH. But what we're interested in is this guy, luteinizing hormone. So luteinizing hormone peaks, and that is what causes ovulation. And when ovulation occurs, what are the granulosa cells going to do? Remember, the granulosa cells are going to become corpus luteum. And take a look at the estrogen levels. Estrogen levels start to drop, and all of a sudden, we start to get progesterone levels rising. So that's all due to changing over our gears into progesterone dominance at that stage, which is important for maintaining the uterine lining. We'll stop there and fill in notes later on.